My name is Nihal and my class number is 53. Today we will discuss glucose transporters. So to begin, first of all, why do we need glucose transporters? So glucose is hydrophilic, as we can see that glucose in glucose structure, it has many hydroxyl groups. So it make it very hydrophilic since polar molecules cannot be transported across the plasma membrane carrier. So the carrier proteins called glucose transporters are needed for cellular uptake. There are two types of transporters, GLUTs or glucose transporters and sodium dependent glucose transporters. So actually we have 14 different types of glucose transporters. They are found throughout the body in all cell types, and the glucose transporters transport glucose across the plasma membrane by means of a facilitated diffusion mechanism. There are 14 different types, and they are sodium and ATP independent. They do not require sodium or ATP, and the word ubiquitous means that they are transported and they are present all over the body. And sodium dependent glucose transporters are sodium and ATP dependent, and they are not ubiquitous. They are only located in intestine, renal tubules, blood brain barriers. So to begin with glucose transporters, there are actually five most important glucose transporters. So the important glucose transporters include glucose transporter 1, 2, 3, 4 and 5. The rest of the transporters may be important, but their significance is not yet discovered. Glucose transporter 1, so it is found in blood-brain barrier, heart, fetus, and it is insulin independent. It is the most ubiquitous among the five important glucose transporters, and it is first glucose transporter to be cloned, and our all tissues, many cells of the adult contain it, and all tissues of the fetus contain this transporter. According to the research, the amino acid sequence present in glucose transporter 1 in human is similar 98% to rat and 97% to mouse, rabbit, pig, and it means that it is functionally very important. So the second important glucose transporter is glucose transporter 2, and it is present in kidney, liver, pancreas and small intestine. It is insulin independent and it has high Km. So what is Km? Km is actually Michel's constant. An enzyme with a high Km has low affinity for its structure. In biochemistry, Km stands for Michel's constant, and if an enzyme has high Km, so it has low affinity towards its substrate, and its substrate must be present in high concentration in order to achieve maximum concentration or maximum affinity. So it is obvious that liver cannot take every time blood glucose, and liver only take glucose when liver kidney only take glucose when it is present at a high amount. As we know that in our body there is an gland known as pancreas, and pancreas is both endocrine and exocrine gland, and beta cells are present in pancreas. So the glucose level is monitored by beta cells of pancreas, and glucose enter to beta cells by a glucose 2 transporter. Inside the beta cell, it is metabolized to produce ATP. When there is high concentration of glucose, high amount of ATP will be produced, and hence ATP will bind to ATP sensitive potassium channel, and it will cause depolarization and will close potassium channels. Calcium voltage gated channel will open, and calcium enter the beta cells and causes exocytosis, and insulin enters the blood and travels throughout the body, and when insulin travels throughout the body, what it will do that it will bind on insulin receptors present on muscles or fat cells, and on muscles and fat cells there is glucose transporter 4, and what it will do that it will allow the blood glucose to enter into the muscle and fat cells, and so the glycogenesis process can happen there. So glucose 2 is very important for stimulation of insulin secretion. So the third important glucose transporter is glucose transporter 3. It is present in brains, neuron, kidney, and it is insulin independent, and it has low Michaelis constant and high affinity of blood glucose. As our brain cells require energy in the form of glucose every time, it is not dependent on the amount of glucose. Even if there is a very low amount of glucose, it will be traveled to the brain cells. It is not highly conserved among other species such as glucose transporter 1, as the amino acid sequence of glucose transporter 1 was very similar to the raised rats and mouse. It is not similar like that. Major neuron glucose transporter, and it is considered as major neuronal glucose transporter as it is present in both dendrites and axons, and it is ubiquitous, it is found ubiquitous in our body, but its function is not discovered yet, that what important function they perform there. And in rats, it is found to be only present in glucose transporter 3 is only found to be present in brain. So the fourth glucose transporter is glucose transporter 4. It is present in muscle, adipose tissues, and heart, and it is insulin dependent. And it is the only glucose transporter which is insulin dependent. It has moderate Km, Michaelis constant, and moderate affinity for glucose. And the most important thing about glucose transporter 4 it is that it is insulin dependent. The way we study in glucose transporter 2, and when we study that when insulin was entered into the blood, so then what it did, that it binded to insulin receptor present on muscles, and then it allowed glucose transporter 4 to be activated, and then it allowed glucose to leave the blood and enter the muscles and fat cells, and then glucogenesis will occur. So it is insulin dependent, and according to the research, it is 95, its amino acid sequence is 95% similar to the rat and 96% to the mouse. The fifth glucose transporter is glucose transporter 5. It is present in intestinal epithelium, and it is insulin independent, and it is major fructose transporter. It is high fructose specific, and fructose is transported across intestinal epithelial cells by passive transport through GLUT5. Passive transport means that it do not require energy the way active transport requires, and it is also found in other parts such as kidney, brain, muscles, adipose tissues, but in these tissues its significance is not yet discovered. Now we will study sodium dependent glucose transporters. As the names suggest that they are sodium dependent, and they require ATP. So the first one is sodium glucose transporter 1. It is present in interior side of intestinal epithelium, and it is insulin independent and sodium and ATP dependent, and its major role is glucose absorption. So intestinal SGLT1 is actively transport glucose and water into the intestines against its concentration gradient. It is active transport, and it uses energy generated by sodium and electrical gradients. SGL2 is the second sodium dependent glucose transporter, and it is independent of insulin, and it is sodium and ATP dependent, and its main role is in glucose retention. In kidney plays a main role in glucose homeostasis by regulating gluconeogenesis, and renal glucose reabsorption happens 90% in SGLT2 and 10% in SGLT1. So this is one of the very important transporter, and in order to be healthy and don't have conditions like glycosuria, so we must have these transporters actively functioning.